Good afternoon, Dr. Ravishankar.
Yeah, good afternoon. Am I audible?
Yes, Doctor.
Yeah.
Yes. Students, we are entering into the last session of the day. We have with us for this session Dr. Ravishankar, Principal Scientist and Coordinator, Studio of Computational Biology and Bioinformatics, the Himalayan Center for High Throughput Computational Biology from the CSIR Institute of Himalayan Bioresource Technology, Palapur, Himachal Pradesh. Dr. Ravishankar has his BSc from University of Delhi, MSc Biochemistry from Jamia Hamad, New Delhi, postgraduate advanced diploma in bioinformatics, JNU, and a PhD bioinformatics, biotechnology, IGIB, CSIR, Delhi. He has a professional and research experiences. A researcher worked as a researcher at Genetic Information Research Institute, GIRI, California, USA. As a main bioinformatics in BioBase International, Bangalore, and as a faculty in bioinformatics in Indian Institute of Advanced Research, Gandhinagar, Gujarat. His area of interests are regulatory system analysis with respect to microRNAs, computational epigenomics and regulomics, computational issues with next generation sequencing. He has guided 8 PhD students so far. He has a few fellowships and recognitions like GATE qualified, CSR NET qualified, DBT fellowship, Helvet Packard fellowship, NIH USA project fellowship, DST SERB Young Scientist Fast Track Grant Award, member Board of Examiners, Central University of Bihar, INSA Australian Academy of Science Visiting Scientist Fellowship 2012-13, and as visiting faculty and CDC committee member, Central University of Himachal Pradesh. He is also serving as an academic referee and reviewer on top tier journals like Nucleic Acid Research, FEBS, RNA from Cold Spring Harbor Lab Publications, Nature Scientific Reports, BMC Bioinformatics, CRC Press, BMC Genomics, Genomics, Proteomics and Bioinformatics, Genome, PLOS 1, Genomics from Elsevier, Database from Oxford, Plant Molecular Biology, Journal of Bioscience, Journal of Genetics, Molecular Breeding, and PeerJ. He has so far secured a fund for 10 projects as a principal investigator from several funding agencies, and are also served as a co-PI in several projects. He has a publication of 45 plus publications in international reputed journals with an H index of 17. So with this short introduction, I invite Dr. Ravishankar to deliver his talk. Doctor, the floor is yours.
Yeah, thank you very much. It was actually very long introduction. My introduction is just simple. I'm a student of bioinformatics, Ravishankar. Okay, and I share my screen here, and just a minute. Presentation, I just share it here. Can you see my desktop?
Yes, doctor.
Okay. So, just a minute. So my today's topic of this lecture is that how AI eyes the genomic world. You must be very much aware by now that artificial intelligence, it is too much talked topic, and you keep on hearing about it. Everybody is doing some sort of data science course, paying several lakhs, even up to several lakhs for 6-month training even. But my big concern is that if you don't know anything or you have not been aware for it, does not mean that it has just taken birth today itself. So actually in this field of bioinformatics, we have been practicing this AI, artificial intelligence, or say machine learning, for a very long time. I was student of bioinformatics, and I started studying bioinformatics in year 2000 when the human genome first draft was reported. And before me, for 5-6 years, or even before that, many people were already in abroad were studying bioinformatics, and these all neural network models, all these things were being implemented since 1980s itself. So why it is so popular now? The question is that, and the answer is that due to the advent of this mobile technology and facial recognition, pictorial recognition, classification, all these things. And the second thing is political thing, that if, somehow they learn that they can read your Facebook pages, and they can assess your emotions using machine learning algorithms. So using that, many election trends are set, and election were won in 1970s itself. Sorry, it's perhaps in 70s or 80s, I don't remember exactly. In USA itself, this thing was had started using machine learning to win elections. And in India, 2014 election was won by using machine learning itself. So it is nothing new, actually. It has been happening, but awareness was lesser. Now you guys are aware that what is all artificial intelligence is about. So this is my lab. Here I stay, and I have been running a laboratory for past 12 years. And this is uh, the Studio of Computational Biology and Bioinformatics, also a Bioinformatics Center of DBT. And so let's start with first thing, that bioinformatics. I specialize in bioinformatics. So how you take approach this subject actually? So a very important component of this entire science is computer, and you need to know about how computer operates, because as a new student of this subject, I faced very much difficulty. I had a background in biochemistry, and I was knowing nothing about computers at that time. And n- that is very essential thing now. So why computer, and how your first query will be how computer can solve or look into biological system. That is very peculiar thing, because computer looks different thing. You can watch video on that, play games on that, you can prepare MS documents, but biology, that is impossible. So when I was actually joining this bioinformatics course in 2000, some of my seniors and teachers, they suggested me, oh, where you are going, computers are all about games and all these things. So that kind of awareness at that time it was. The computer, why it is important, and electricity, how it is associated with computers so much. uh, Thing is that whatever computer does, it does very simply actually. Yes and no, yes, no. So you can see here, this all thing, this is no, this is yes. Okay. So everything goes into between these two, 0 and 1. So if anything has to be read, it is converted into 0 and 1 binary digits. And finally, this yes and no, the way this all decisions are made by computer is through the logical gates. So logic gates, you must have studied about logic gates in your 11th or 12th classes. So these are like yes, and how to arrange yes and low, no, and then what will be the output. So there are many kinds of logic gates, like and, or, zor, not, nand, and nor. So and means that something is happening. If A is there and B is there, then output will be 1. If A is there or B is there, in दोनों में से कोई भी होगा. My just one request is there. If anybody has problem with English, my English, then I can come to Hindi also. I know only 2 language, Hindi और English. तो जिसको भी पूछना हो हिंदी में भी वो पूछ सकता है. ठीक है? I am comfortable with Hindi also. I'm much comfortable actually. So and this, or if something is coming from A side, okay, just a minute.
Doctor, we are losing your voice.
Am I audible right now?
The voice is very feeble.
Just a minute. Please give me a moment. Now it is fine.
Yes, yes.
So suppose something is there. This is A and this is B. If A is giving signal of 1 and B is giving 0, then output will be there 1. That is OR. Anyways, any one of them should click. So that is OR. Then you have XOR, exclusively OR. Any one of them should have. Exclusively OR means that if one is, this one A is having input of A and B is having input of 0, then it will be okay. 1 will be the output. If it is 1 here, okay, if it is 1 here and it is also 1 here, okay, then output will be 0. So that is the XOR gate. Then NOT is something like that, that if input is 1, then output will be 0. जो होगा उसका उल्टा होगा. If whatever is the signal, the opposite thing you will get. So if you say yes, the output will say no. So it is like that. Then NAND, opposite of AND gate, that whatever was the output of AND gate, AND means that the input should come 1 1 from both sides, then only 1 will happen. But in NAND, that thing goes opposite, and input from both sides is, if 1 1, then output will be 0, no. Same way, NOR, opposite of OR. And these things are defined in computers by their truth tables. That is same thing what I am saying you, that that is kind of frame in which yes, no, and then yes, no, all these things are defined. So these all computers are all about these logic gates, how they are arranged, how these logic gates, millions of these, thousands of these logic gates are applied, and the computer chip, the processor is designed. So what the thing you see, the processor, it is actually a collection of these this element, lots of these, and that is called transistor. So you see the transistor has two kinds of things. This is kind of triode, input, output, and base, ground. So this ground, what it does, that it controls the flow of electricity. If it does not control the flow of electricity, the electricity will not flow finally, and everything will be stopped. So yes, no signal is very minutely controlled by transistor, and this all truth table is actually implemented 0 1 0 1. So 0, flow will be not there in the electricity. 1, the electricity will be there. So these kind of transistors are used rampantly, and large number of integrations are done to prepare the circuits. So these are the pictorial representation, how they are working, and how these transistors are seen. I'm just showing you here. So these all the circuits have been classified in processor like as a small scale integration. 1 to 10 gates are there, logic gates are there. Medium scale integration, here you have 10 to 100. Then you have 100 to lakh, kind of. More than lakh is VLSI, very large scale integration. So these are the classification of actually processor chips. So all present date processor are in the last category, actually this one. And the man behind this all is the George Bull. George Bull was a small kid from a very poor family, and he used to work as a shoemaker, and he was very much inclined toward study maths and all these things, logical thing, and he was an avid reader. So he taught himself, and thereafter, after some time, he joined a school where he used to taught and study also. And thereafter he wrote a very interesting book, Introduction into the Laws of Thoughts, how we think. That book was written, and then he founded the new area of mathematics, that is Boolean algebra, and that how these all logic gates 0 1, how you do counting on 1 2 3 binary decimal system, but he did all things on 0 and 1. So how all calculations are done on 0 and 1 was shown by George Bull. And after many years of him, like in 1945, we saw the proper computer. So you see, almost 100 years later, computer arrived, but his logic was very much there itself. And who was knowing that his logic was going to revolutionize entire world this way, which we see today itself. So foundation of living world now. So next question is that, okay, so 0101 is there. So how you see words document there, PDF files there, many things are going on inside the computer. So computer, how it sees that? So that is encoding actually. So like I'm just showing you the very basic thing, like on your keyboard you see that A B C D E F G H characters are there, and then tab, all the special characters are there, plus minus signs. So initially they all these were uh, encoded into ASCII codes. Okay. So just a minute. So here you see ASCII code. This ASCII code was the conversion actually. So here you are seeing how many position: first, second, third, fourth, fifth, sixth, seven. So you call it 7-bit system. Earlier it was 7-bit system. Later on it became 8-bit system. Okay, 8 bits. So what it is doing actually? So they found that all these characters could be represented like, suppose if I consider 1 bit is like one shell, one place. So here what, how many values you can write? Either it will be 0 or it will be 1. So same way, all these combinations, if you use, then 7 positions or 8 positions combination can cover all these characters, and they can be encoded into unique numbers using just binary digits. Okay. So that from the here everything started actually. So later on it became 8 characters. So you can simply see that like here you have two kinds of watches. One is analog watch and another one is digital watch. So transformation is the what you say, the foundation of this entire computer system and data analysis. So everything has to be transformed into a doable form, and everything is almost done same way. So here you see the computer system works with the fundamentals of 2 to the power n. So since it is binary digit based, so everything you see is as a multiple of 2 basically. So RAM you get either 1 GB, 2 GB, 4 GB, 128 GB. And computer architecture are defined as 32-bit system or 62-bit system. The bit system I'm telling you here because this 32 these days we are working on 62-bit system or even higher one actually. So what happens that all this communication is done in 32 bits. Earlier you were seeing 7 bits or 8-bit system, 8-bit system actually. So in computation starting was done with ASCII characters, 8-bit system. Okay, all these A B C D X Y Z character plus minus, all these could be covered by 256 uh, uh, numbers. These 256 numbers could be represented using 8-bit system. Okay, 8-bit system, any 8 bits binary digits. And later on now we have other fonts like Chinese, Hindi, all these things, so they have to accommodate it. So now we have bigger system, 64-bit system is there, and instead of ASCII you work with Unicode. So I will be coming to that part also. So this is now ASCII was refined further, and then you can see here that 126 you just a minute, my mouse goes very small when it comes on screen, so I don't find it easily. So up to 127 characters you can see here, this part were covered, and counting starts in computer with 0, not from 1. So 128 and plus minus values total make out 256 characters. In earlier time there were met possibilities, the text or character things which could be formed or special characters. And later on various other languages came, other applications came, so it expanded. So how actually numbers are converted into binary digits, you can see here that suppose if I have a number 943 here, then how it is calculated. 3 is written here, it is multiplied by 10 to the power 0. In the decimal system I'm showing you how you are doing actually thing. So 3 can be written at the first place, 3 can be written 10 to the power 0, so it becomes 3 because 10 to the power 0 is equal to 1. Then you have 10 to the power 1, so that is 10. The 10th position, that is 40. So it is like that you are adding 40 plus 3 and plus this is 10 to the power 2. Okay, this is 0th position, this is first position, this is second position. The same way you have to just put the power on 10, and you see here that is 900. So it sums up to 943 here. So similar way decimal the binary thing is done. So just you see here, uh, if you make the base of 13 here, the same way 13 to the power 0, 13 to the power 1, 13 to the power 2, and the number is formed. So binary, same way it is shown here, that how you solve the binary numbers. And you see the base is here written, the 2. So you just do it 2 to the power 0, 2 to the power. So if some number, binary number is written here, you just do like that, 2 to the power 0 multiplied by 1. Second position it is 0, and base is 2 to the power, the first position it is there, so you write 1, then 2 to the power 2, and same way you keep on doing the same thing, and you finally get the decimal number of 233. So how decimal number are converted to binary? It is just suppose you keep on dividing the number by 2, and you in computers you write from right to left. So it is 1 here, then you again do the division by 2. You keep on dividing until the last position, and you get the binary number accordingly. I'm just showing you the one basic thing, that how these binary numbers are operated. So like 0 is shown by all when all bulbs are not glowing. Okay, 0 is there. So all 4 positions, suppose I'm just taking a 4-bit system. Okay, first position, second position, third position, and fourth position. So it is a 4-bit system. So every position is being assigned 0 and 1 values. So to represent 1, all positions are 0. Then to represent 1, first position was given 0. So first position was given 1, and second position was given 0. And then you what happens, then you come to this point 2, then 0 is shifted to this part, and it becomes 0. Then next both position become 0. So what you do now, just look here. If you sum 2 and 1, it should come to 3. So what happens, 2 and 1 comes to 3, how? You add 1 to 0, it becomes 1. And here also you add 0 to 1, it becomes 1. But what happens if 1 and 1 are added? So what happens if I add like this one 3, this is 4, and this is 5, and this is 6. Okay, so what will happen if I add this into this one? So 3 plus 1. So that is what happens in binary algebra, that if you add 1 plus 1, then the position becomes 0, and the 1 is carried forward to the next position. So same thing here happens, that if you add here, you can see that if you add 3 and 1, then what happens? It should become 4. And what happens? 1 goes to this. Just a minute. Just a minute. Just a minute. हाँ अश्विनी, हाँ मैं अभी मीटिंग हूँ, मैं थोड़ी देर में आपको कॉल करता हूँ. So 1 is carried forward here. Then what you see here, that this one is already having 1. So this one is carried forward here, and it becomes 4. So the same way all additions are done, and similar fashion the subtraction is done. So they have a different kind of treatment. So So this is all about the Boolean algebra, how it works basically. So I told you that Unicode system was done because earlier 256 characters were there in a sky, but as we expanded, we started processing digital photographs, and then uh, the communication system expanded. So we expanded to higher level of bits, and Unicode was formed. So present system of all computers work with Unicode. I'm just showing that how important it is, thing like that transformation of anything into data. Anything can be transformed into values, numbers, and digits, all these things. Okay, so computers can work only thereafter. How they do? The best example is that you must be knowing that today you know about your smartphones. So you'll go for this megapixel, how many megapixel they are providing. Well, you know, pixel is what? Pixel are like small bins. So if you see here, so suppose if you concentrate on this region or pick any region, you see here, you can see small square shaped areas. So the actually a picture is a aggregation of lots of square shaped areas, and they come together, form a sort of matrix. And for every such squared area position, you give value for red, green, and blue color. So red, green, blue, how much proportion they are having. So their proportion up to 255. So you, if you go to the setting of your color pixels in your computers, they give you option RGB, how many bit character based, 8-bit character based, or 16 bit, or 64-bit, something like that they ask. And these values, you can see that red is converted in from 0 to 255 values. Similarly green. So whatever value you put here, the corresponding value is then converted into binary form, and accordingly that pixel is color is defined. Whatever color you mix of R and G and blue, the same kind combination is just represented there. So accordingly the color coding is done and picture is defined. So RGB is, that's why important. So when you see RGB, that means for a given location in the matrix, you are going to fill this value mathematically. These values are basically converted into binary digits. Okay, and then binary digits are operated, and that you see the appearance on the screen that comes based on that calculation only. So finally coming to your part, that foundation of AI. So what actually runs AI? So up to this time you must have understood the importance of uh, numbers, that how numbers are important in computers. And basically numbers, everything can be converted in anything. My voice can be converted into numeric form. Image use, just in the last example, previous example, I showed you that how the image is converted into numeric form. So everything in this world can be converted into numeric form. Okay, and then if something is converted into numeric form, then all calculations can be done. So same thing happens. So foundation of artificial intelligence happened that with why you are calling it artificial intelligence. Artificial intelligence is that something which you used to decide, now your computer will be able to decide it. Okay, so you wanted to automate several things. And in biology, it is very much essential, because you are not going to end up with just one experiment that is very rational system that used to be in previous generation. But now you are getting lots of data, and this system, they work, in what you say, in very much associated manner, very much combined way they work. And they define a particular condition of cell system, at what kind of tissue they are going to form, what kind of response the cell is going to have if it gets cold, temperature difference, or stress sort of things, or disease. That is key, that keeps on changing drastically and minutely at every level, and everything is going to define the change. So in that scenario, you need to deal with lots of equations at the same time. Lots of different data are needed, and then you are going to understand anything fully. Otherwise you are not going to find anything completely. So basically you have to take decisions. So 0 1, you saw that how computer took decision in the logic gates, 0 and 1, 1 and 0. The entire life is full of that 0 and 1 decision kind of things. So everything runs between this 0 and 1 actually. So in your 11th class, if I remember, in coordinate geometry, they taught you the equation of line. So the very same thing is there. So here you can see here y equal to fx. So there you used to be taught like y equal to mx plus c, and same is the line of regression. So all story starts from here actually, line of regression. So if you find something here like that, here I have to compare how much height is related to weight. Can height of a person tell about the weight of the person? So you are just trying to fit that line, and you find out the best optimum line by get minimizing the error is that how much the points differ from the or deflect from the particular uh, line. So that is the error. You try to minimize that error. So sometimes you move left, sometimes you move right. You try to minimize that error, and then you say that yeah, this is the line of regression which fits the best to the given data points. Okay. And what happens in classification? In classification, you just define a separate boundary that yeah, I had this line and it defined various data points, and if something goes below this line, then it will belong to this class, and if it goes above that line, it will belong to different class, so second class. So that is the difference between regression and classification. These two things are very much in the machine learning. Basically machine learning is mainly about classification and regression. Regression gives value that if height is this, if something is that the input data is this, then what will be the outcome is given by regression. And here is the question that if input data is this, then to which class it is going to belong, to this class or to this upper one class. Okay, so that is the difference between regression and uh, uh, classification. Application of artificial intelligence is not just like that regression as VM. Another thing I have classified them into this one, supervised learning. So regression and classification, they come into supervised learning. And for unsupervised learning, you can see here that it comes like clustering. Okay, so clustering is kind of unsupervised. Here what happens, supervised learning, what happens, that you have a data set already known. You are knowing that this kind of numbers are there, they belong to some are in positive class, some are in negative class. So you know the level, so they make your training set. You train them. Okay, you tell the machine that see, I am training you, and if you find this kind of sequence, it is plus. If you find this kind of sequence, minus. So let me know that how well you learn here. So you keep on training and you keep on testing that if the training was successful and error was minimized or not here, just like that. So here it is not the same in unsupervised learning condition. Just so uh, so in unsupervised learning, I just apologize because my mouse is so small here, na, it is not showing the screen properly. So in the unsupervised learning here, the thing is not like that. You are not giving any label. So here it was matter of label. Here in that case, you are giving label minus plus the class category you are defining. Suppose if the person is male or if the person is female, you are classifying like that. But here it is not like you are not knowing about the labels. So what happens here, the data points, they they start calculating difference between each other, okay, simultaneously, and based on their proximity. for a given value, they form cluster kind of things. So they form groups on of themselves. So but you don't know their class labels. So thereafter, once they form some kind of group, then you pick them, you try to classify them. Thereafter you try to learn that what kind of uh, genes they are, like cell types or gene types. Suppose I have particular gene here, I have taken expression, gene expression and gene expression 2. So here I am trying to, based on these two values, I am trying to cluster a group these sequences. So every sequence is showing some kind of proximity in this group based on this kind of relationship between the expression of these two. So in actual condition, lots of values come into the process of clustering. So basically in machine learning, finally you have this point here, that you have to reduce bias and variance and keep the balance. These are two weaknesses of any machine learning problem: bias and variance. So what is bias and variance? Variance is how much a performance changes with training data. So I told you here that I was giving training data here. So on that data, the machine learns, and then a new fresh data is given to it, that is test data. Test. So test data is provided, and then on that test data, the machine capability is tested, that how good it did. So variance is that if you provide data and then you change the data and provide a fresh set of data, then the program, the machine learning model, that may not perform very good. So in actual learning data, it worked very well, but when working on the new data, it shows lots of variance in performance. So that is called variance change, an inability to accurately capture the performance on the fresh set of data. A bias is that innate problem with the machine learning algorithm. What kind of function you are using? Like here, I told you that simple line function was used, but it is not like that. When you start, that is a very basic point. When you look into machine learning part, lots of different kind of functions are there, and that is the purpose, that what kind of function is going to be fitted there. So kind of function being used, that function has a limitation. So it is not going to perform beyond a particular threshold. Okay, so that problem it will face. So bias happens like that, due to that kind of functions. So you try to balance between this variance and bias all the time. So the complex what is a complex functions, the complex functions have high variance. So if you show them new data, they perform bad. Okay, in old data they perform good because they learned very well, and they try to capture every point. But on new data they fail. Here what happens, that whatever data you provide, they perform almost reasonably same level, they don't show overfitting, but beyond a particular limit they can't go. And third point is variance of error. Error is like that, c here, y equal to mx plus c, that innate problem, the innate weakness of the learning algorithm. Like I am showing you here the plot. So whatever you plot here, okay, c will be there. So cut like a line peg. So that is going to happen. So that is innate problem, that much amount of error is certainly going to happen, and you can't reduce this one. It can't be reduced. So that error is going to be there. So entire machine learning is about all this settlement between variance and bias. How you adjust and balance variance and bias and get the best performance is the purpose of machine learning and training and all this AI stuff. So I just show you here same thing, like y equal to mx plus c. So earlier I was just giving you one variable. Here you have got lots of variables. So what you try to do, that if in that example the height was one variable, but you can have many variables now together here, three and number of times, and every variable is weighted here. So beta n, beta 3, beta 4, whatever you see here, these are the weights. And even regression coefficient you call in terms of regression. So that is the main purpose of machine learning, that training in during the training you try to adjust all these weights and arrive to a function which gives you the best values. Okay, so that is the thing. Sometimes we keep on shifting this. Just a minute, I change the color. So you can see that if I move the line here, in order to do that I have to change these values. Okay, beta values, when you change them, then the slope of the line changes and it keeps moving. So this is how it is done, and all these values of x1, x2, x3, xn, these are actually what? These are actually the values of variables which we call features. So if for every data points, like I have a sample of 100%, I have to decide that whether the person is man or woman. Then I get the data point one, like one man, then another person, another fellow, third fellow, fourth fellow, and fellow. So I that is a set, that is the training set on which I keep doing studying and training the learning model. Okay, and based on that, then another set is there, the testing set, on which I test. All of them are defined across these variables which are called features. Without features you can't perform any machine learning or artificial intelligence work. So you need to provide input basis of on which basis I can distinguish between these two classes category or multiple classes category. What are that is very much important thing. So I just explained how it becomes important. You have got you have to characterize between a monkey and human. So monkey and human, if uh, how to characterize that thing. So these are the features I am looking for. Okay, so here limb size, eye diameter, jumping height, constructivity, how good thing they can make. Based on these, I populate the values for human. Okay, whatever values are v1, v2, whatever, 10, 20. So these, like x1 limb size, I should not put 0 because that is amputated guy then. So limb size is suppose 3 feet. Okay, something like that. Similarly you put the values here, and what you find that you get that complete training data set for human and monkey like that, and then you know the level, and based on that you first train and then you test. Both thing you do. And there are some features. The main thing is that how much importance the machine should give to a feature. So how much weight, that is the beta. We in the previous slide I showed you that the beta value. So accordingly the features are weighted. Most important features are weighted more, and then they're based on their discriminatory property, and this process is called feature selection process also. So feature selection and data processing are two major building blocks of machine learning or API. So data processing, like you collect the data, replace input, remove outliers, and duplicate bad examples are removed. Data are partitioned into training and testing data sets. You normalize the data, because in previous example I showed you that one or something like 3 feet or these kind of value, but in actual the values in machine learning are usually between 0 to 1. So that is called normalization. You try to put all these values between 0 to 1 actually, or if you don't do, then they are automated system, they take care of that, but all computations are done between 0 to 1 actually. And sometimes it is pure and pure binary, so either 0 or 1, it is like that. So these were data are full, but the maximum limit is 1 most of the time. Scale, normalize, standardize, unbias or balance, and augment strengthen the data. Similarly here in feature selection, there is entire component called feature engineering actually. So you try to identify the best features. So if I have to identify, I have to distinguish between human and monkey, then I have to somehow find out the best possible features. If I don't select good features, I'm not going to prepare a good model, the machine will be unable to learn. What I do that when I see a monkey, then monkey's face is different than human. Then I have to put values how to define monkey's face. Okay, monkey face has round eyes. Okay, the hair is not there, and some V-shaped head is there. The hair coming in the V-shaped manner, and mouth is lacking lips, proper lips projections. Nose is not projected. Okay, so nose height I can put there, and these kind of things can define monkey's face. Yeah, one feature is there, the tail. That you don't need to any give any value for that one. Just put 0 or 1. Tail is there, 1, yes, or no, 0. Just based on this feature alone, you will be able to discriminate between monkey and human. But what about the condition where you have got even guy bhans like thing? Okay, and you have to distinguish between human, guy, bhans, chimpanzee, lemur, langurs, then things will be complicated. So you see that even face reading AI, they all are human. Everybody is using mobile app. Okay, then how face lock recognizes that yes, this man is my owner? So accordingly, the mobile unlocks itself. So that is the thing. The more complicated situation in real world actually exists. So it is not like distinguishing between monkey and a human. In actual, there are lots of choices, and there from there those choices, it has to perfectly identify from every angle that yes, this is the right choice. Okay, so feature is, that's why important how good feature you are having. Okay, so you have to select that thing, or then if you are not able to select, then for complicated features there is most advanced machine learning algorithm that is called deep learning, which I will be showing in my next slides. They work actually. So up to this time, I just take sip of water, please. In this slide, I'm showing you that the book of life, which we call DNA, this particular DNA is controlled at very different levels. So transcription is one of the prime level. So if transcription happens, then RNA is formed. Okay, this one. So this entire process is very much intertwined. Lots of factors are involved, and from RNA you get proteins, and they control different things. In our genomic system, like in human, only 3%. Okay, only 3% maximum code for proteins. Rest of the genome is not coded, and what I understand that more in our most of our schools and college education, we are only taught about genes and all these things, but system does not work like that. There are lots of things uh, which are beyond protein coding things, and protein codings are just like a certain group of components of our living system, which which are used like tools by the genomic system and cell system to maintain a living system. Okay, so how you can see here now, RBPs are involved also. So in the previous slide, I showed you here the transcription process, and here what happens that even if RNA is formed, RNA can't survive alone. So my most of the, I'm taking context from my study itself, whatever I study. So my most of the work here, as you have heard, is based on regulatory system analysis, how all these proteins are formed, how genes are formed, how genes are expressed, what controls what, all these relationship I look into all these factors. So this is just a small snapshot. Actually, the entire system is so much uh, complex that you can't view them by naked eyes. They like they are highly nano integrated circuits. Okay, and everybody is speaking with somebody. Okay, interaction is going on. So I am just showing you that one component transcription I studied, and transcription different in transcription. It is not like protein coding genes, but there are many classes of RNAs like pRNA, microRNAs, messenger RNA, long non-coding RNAs, no RNAs. They're very different kinds of RNAs are formed, and they have different functions and roles. and no RNA can survive in the cell system without associating itself with some protein. So if RNA is there, then a protein is must to accompany it. If otherwise the RNA is degraded. So RNA protein interaction, we had to study that thing. And why we were studying, we were focusing actually upon this particular microRNA biogenesis factor. We were studying this part, and we felt that microRNA biogenesis, these are the microRNAs are small RNAs actually, and they what they do that they bind to RNA and stop the process of translation. The gene stop making protein, so that is required. The very important component they are. They are almost regulating every gene. Almost every gene are being regulated by microRNAs. But the knowledge about microRNA biogenesis was not very clear. So we try to look into the thing that when which microRNA, there are more than 2,000 microRNAs in human system. uh, and in our analysis tells that there are more than 11,000 microRNAs actually working inside human system. And all of them are very specific. At particular time they come, they do their job, and they vanish. They just gayab ho jate hain. Okay, then another set of microRNA come, they do their duty. Their different combination define different fate for the cell system. So we had to capture all these things, that how these micro different kind of microRNAs are being formed. Okay, and that study I had to do. And for that we had certain kind of data also. But all these things had to be converted into mathematical form and form the machine learning models. Before going into that part, I just tell you that in this slide I'm just trying to show you that how much important artificial intelligence and computation has been in the biological science. So human genome was decoded in this year finally, 2000, year 2000. Okay, here it is written here. But when it had started, when it had started here, it had started here, 1984, 1984 से ये काम start हुआ, and it ended here in 2000. First draft of human genome was released in year 2000. The very same year I had joined studying bioinformatics, encouraged by this report. Why I got so much encouraged, fascinated by this subject, bioinformatics? So I tell you here, इतना ज़्यादा समय 1984 से काम स्टार्ट हुआ और इतना साल लग गया. So what was happening that they were sequencing genome. So genome was like that मैगी का नूडल, एक endless नूडल की तरह उलझा हुआ आपस में, all intertwined. And the you can't sequence a genome just like that कि एक genome का line लिया and you start reading it like this and you report that this is the sequence. It does not happen. There are limitations. The chemistry involved that does not allow reading beyond a particular limit length. So these are the length. They are called reads. Okay, reads. These reads were formed, and then these reads were cloned into back libraries, bacterial libraries. These are sort of okay. So why they are cloned in back libraries? Because multiplication of this bacterial growths are very much uh, high, so they multiply very fast. So lots of copies are formed. When high number of copies are formed, then you get multiple evidences. Yeah, this region is like that. Okay, so that's why multiple libraries are formed. So thereafter they are finally broken further and then sequenced again for their multiple copies. Their final short reads are there, and then they are again tried to be assembled. So this is connected to this one, this is connected to this one, so you call that this region belong to actually in this way. So this is what it is happening here. You got these short reads. One read reads like that, another read reads like that. You find this overlap between these two here. Overlap is there. Okay, they are matching, so they are together. So if this one ends here, this one starts here, so join them. and then you get in turn a longer sequence. That is called assembly, and that is genome assembly. I told you, you heard it. It is very simple, but it is actually not that simple. Lots of complications are there. Our eukaryotic genomes are full of repetitive sequences actually. Okay, repeats. 3% is only unique region, coding region. Na? I told you there in the last slide. And the rest are repetitive or non-coding regions. They make us a complicated human being. Okay, we are not bacteria. That's why. And all the plants you see, they are much complicated than us. They are more evolved and complex than us. The plants, they are having lots of repeats. Repeats means all are looking similar, like this region similar. So suppose you have this unique region here, and I just change this color. So you say that up to this point it was unique, and after that you get here the repeat region. Okay, yeah, this is repeat region, this is repeat region, and this is repeat region here. If this region is also same, this region is also same, this region is also same, and for assembling you need to look for matching two regions. If all more than two these matching regions, you get multiple such regions, then what you will do, what you will end up assembling, it had to assemble to this one, suppose, but you end up assembling with this one. Okay, you do wrong assembly, and that is the thing. So these scientists were not able, they in fact leave it aside, resolving this thing. They were not even able to join these two, because by wet biologists, the experimental biologists, they can sequence, read, but they don't know how to join things. So they were literally doing manual things. स्मॉल पढ़ रहे थे, लेके 1984 से लेके पढ़ते ही जा रहे थे. ओके, यहाँ से ये देखो, ये तब से पढ़ते ही जा रहे थे, और उनको 2016 ईयर्स दे टुक टू रीच assemble. And even वो भी नहीं होता, even they would not be doing that part. Okay, 5 countries were involved: USA, Europe, and Japan. Lots of countries were actually indirectly also involved there. Okay, in human genome project, but they were unable to solve genome, just due to this small step. Manually they were reading and joining, manually reading and joining, and they were ending up nowhere. In 1990s, end of the 19 and early 19 and the early 2000, or say end of the 1999, something like that, a young guy from Cella Genomics, okay, he developed a computer program, and the decision which human scientists were taking for past 16 years or 15 years, his simple code could do all the things. So it was a brilliant example of even intelligence by the computer. So that guy that imparted the intelligence to the computer code, he wrote the program. That program looked across all these things, and you got the human genome within 3 weeks. जो 16 years से काम वेट बायोलॉजिस्ट नहीं कर पाए थे, that thing was ended in just 3 weeks. So sequencing at that time was done through Sanger sequencing. It was very costly effort. Everybody could not have Sanger sequencer also. It was very complicated sequencing process also. They had their own share of hardship. Now things have changed. So you can see here that we are now in the era of next generation sequencing. You call it next gen sequencing. That is missing classroom boards. So they have literally revolutionized the entire thing. So now you can, in no time, you can sequence genome. You can find out how proteins are interacting with RNA, how transcription factors and RNA polymerases are setting upon a DNA and transcribing a gene, how long non-coding RNAs are formed. So all these things you can capture just by RNA sequence, this next generation sequencing. And right now you have three major platforms of next generation sequencing, that is Illumina sequencing, that is short read sequencer. So small reads from 36 base to 150 bases you get in Illumina. Okay, PacBio is another sequencer. It is long read. We call it long read sequencer. It generates read size of 5 KB, 5 kilobyte by basis, sorry, and goes up to 20 KB. So for genome sequencing, all these things, this is ideal platform. For RNA sequencing and this protein-protein interaction or microRNA study, this is preferred platform. Oxford Nanopore is cheapest of all, and it gives same kind of 10 KB kind of long reach, almost similar level as this passive biosciences. Oxford, they give long reach. One works through this particular what you say is deuterium cells. I don't pronounce it properly, but it's kind of a lock room is there, and in lock room that immobilized DNA is there, and it is read by the laser guided emission, and the fluorescence is red. Every ATGC is colored by different colored fluorescent uh, attachments, and once some base comes, it releases that fluorescent part. That fluorescent part is detected by the laser, and accordingly the map is formed. Here, here error is little bit higher. What happens that they use cell membrane, and from cell membrane every single nucleotide is passed. Every single nucleotide has its unique what you say pH potential actually. So the pH of the particular once it enters, it changes the pH for a moment. So hydrogen ion concentration that changes, the change is recorded, and accordingly the nucleotide is assigned. So these are the three technologies which can sequence any nucleotide, any protein interaction. They can capture all of these things. I'm just showing one slide here that how this particular what you say RNA-seq is done. So this is the RNA sequencing is done. Then exonic reads you see here. Short reads are found. If you know the RNA sequence, you can simply find the similar regions from where they are coming, and from there you can measure the expression profile, that how much a particular RNA was expressed, the amount, the degree can be calculated. Similarly here, cross-linking studies, like this protein is bound to the RNA. Okay, so here what it is done, that using ultraviolet rays and different kind of chemicals, you immobilize, you lock this protein with the RNA, and what you do then next, you degrade the remaining part of the RNA. This part is not degraded because it got associated with RNA binding protein. Then what you do, then you do immunoprecipitation. So it behaves like an antigen, and antibody is attached to this antigen, and you precipitate it, and then you sequence this region here, and you find that okay, this RNA binding protein was binding to this region of the RNA. Same way here, chip check is there. So for transcription factors, same way you do same kind of things. And the concentration of RNA, the how much RNA is being formed, means that how much a gene is expressing itself, is calculated in RPKM. Okay, you map the the coming reads from various regions of the gene to the gene, and accordingly you quantify them. So what happened? Quantify them. So everything is quantifiable. We quantitate. What happened here? Gene expression जो था वो क्या हो गया? अब यहाँ न्यूमेरिकल वैल्यू में मेरे पास आना शुरू हो गया. Same way, ये count, ये न्यूमेरिकल वैल्यू कहाँ से आ रहा है? From where it is coming? It is coming from all these counts. Here you read mapping. Read map are converted into RPKM values. Okay, and these RPKM values now represent gene expression in terms of numerical value, digits, and I can do wonders with these values. I have no qualm in saying that all these things now transformed, and we use it. How we use it, I am showing you now. So as I told you, I was interested in studying those interaction RNA binding protein, how these RNA binding proteins select their partners where to bind in the RNA. There are so many RNAs, and how they decide where to bind. Okay, so that thing had to be studied. So I had in the back end this ClipSec data, which I showed you here. Okay, this one, this ClipSec data. Okay, these all data were coming from sequencing, and these are available even in public domain. The NCBI and NIH and the GEO, all these places, they keep this data. Every experimental biologist has to report, that is mandatory thing. Their data are reported in SRS, something like that. So from that, there we got all this data. Okay, and what we did, yeah, lots of studies. So what I told you, that machine learning is based on feature selection, features. So my first duty was to identify features, the right features. So lots of statistics were done, motifs, binding motifs. We tried to for, find. We started with a 6 month, यानी 6 bases का एक composition sequence का हम लोग सर्च करना शुरू, जितना every possible 6 months were scanned, and we applied different kind of computational algorithms, and we then expanded them, and from various experimental conditions. What I told you, that you need to have positive data set like man and woman, and negative data set, or vice versa. प्लस को वुमेन भी कर सकते हो और माइनस को मैन भी कर सकते हो, whatever, to distinguish between. So we had data, we had sequences which we are having this protein, RNA binding protein binding to the RNA, and another set negative data was that one where it was not binding. Okay, so we tried to find out the motifs, and we found that certain motifs were discriminatory between positive and negative data sets. So we use them as anchor. Here you can see in this step, we use them as anchor, and then we studied statistically if they are distinguishing between the two data strongly or not. They found the basic feature. Then what we did, the final anchoring was here. One motif are found. Then we found the flanking region information, 75 base pair upside and downside, and we calculated other features. 16 dinucleotides, the combinations of dinucleotides totally form 16 dinucleotide combinations. The pentameric density we evaluated, and heptameric density. My choice for 16 dinucleotide was because the base stacking of RNA or nucleic acid, they depend upon adjacent dinucleotides only. Okay, the structure of RNA very much depend upon the dinucleotides. That's why 16 dinucleotides I opted. And pentamers, the shape of the nucleic acid, what kind of shape it takes, it depends upon pentamer. So pentameric was of my interest, and heptamer was again for further reinforcement of any supportive motifs. So that was the reasoning behind selecting these features, and that's why with this insight, features were determined. Okay, so that's why I told you that if you don't select proper feature, you will fail. And these features were fed into the artificial neural network system, which were having first input layer, two hidden layers, and finally one output layer. So what is the artificial neural network, ANN? The main question is that. So ANN works on your sensory organs, the neurons, how they work. A neuron works when it gets input from other sides. Okay, just my mouse is there. So a neuron works when you have input from other side. It gets input, and then it is fired. It sends signals to the next neuron. Okay, how to behave. Okay, so that communication process keeps on going. So from there, the same thing was adapted. And what happens here, like I shown you the regression equation, same regression equation is almost followed here. Similar kind of regression is followed here. Input is like this, is like dinucleotide density for a particular dinucleotide, for another dinucleotide, some pentamer. Okay, their values are given here. Then a weight is assigned. the beta value in that slide which I had shown you. The weight is assigned, where weight is multiplied actually, and then finally all these values are added and assigned here to x. This x is forwarded to a activation function कि भाई उसको threshold, if it has got threshold or not, to fire. To fire threshold, if it fires, then it predicts the what it is going to assign to this region, and accordingly it is matched to the target, whether it was correct or not. So in actual, the target was this, but the performance was like that. So you measure the difference, what, how much error happened between the actual and the target. Sorry, the target and I wrongly wrote it here. So actually what kind of prediction it made. So the difference is learned, communicated back through back propagation to this artificial neural network system. Okay, and then again the weights are updated. These weights are updated, and again training is done. So that is the role of training data set, and how good the learning was done, then improvement is further made. So next thing was that how uh, these RNA binding proteins are interacting with microRNAs. So here I am showing you, in the previous one I showed you basic thing idea of neural network. So this is called deep neural network because there are two hidden layers. Okay, so it does one regression analysis sort of thing, then another regression analysis sort of thing, another layer does, and more you do, larger number of lines you keep on drawing. So next problem was that microRNAs, how they are being influenced by RNA binding proteins. And I told you that microRNAs are like any other RNA. They are also transcribed. Okay, they don't code for protein. They are further cleaved into a 21 base pair long duplex here. You can see this duplex is finally loaded upon the target RNA. So here you can see that this RNA is there. Okay, and this RNA was getting transcribed and protein was being formed. But once this microRNA arrives here, the process of translation is stopped. The process of formation of protein is stopped, and gene does not form any protein, and it stops. So that is negative regulatory aspect of microRNAs. So that's why I told you that microRNA is very much interesting thing, and knowing that how they are formed, which RNA binding proteins are involved. So in the previous one, what we did, we learned that which RNA binding proteins are binding to which part of RNA, and how I can do that. So same thing now that was done. So on microRNAs, we came arrived with all this binding data, and from this binding data, we formed a sort of Bayesian network, and we, as you can see here, we took lots of experimental data here also. So lots of experimental data where small RNA sequencing data, clip-seq data, and RNA sequencing data. So it gave me gene expression data. This gave me interaction data of between RNA and protein, and this one gave me the short sequence, yani microRNA formation data, and all these things gave us good insight that how these interactions are happening. Based on this, these are the NGS analysis guidelines which we followed, and based on this we formed this particular association models, that which microRNA is being controlled by which RNA binding proteins, and we compared it with the random data also here, and you can see that compared to random data, our association were scoring statistically very high values. These all associations were discovered by using Bayesian algorithm. I don't know if you know Bayesian or not, but it is very primary to probability theory. It is just complementary to basic probability actually. Bayesian. So Bayesian network system we used, and we deciphered all these things. This is another kind of machine learning approach. Here you can see that if I am saying here, association is represented by with deepness in blue color. So suppose this microRNA, MIR181 microRNA, we found various kind of association. You can see here for few I am reporting here. So like here you can see moderate association, and when you come here, very high association is there. For this one, negative association was there. So this kind of matrix representation was there. Based on this association, I could also do one thing. I told you unsupervised learning. I don't have any level here. So what I did unsupervised learning. What I actually did. So these all color codings can be converted into numeric value based on their intensity. So accordingly, these values are converted, and then, like Pythagoras theorem, what you do that you calculate distance between 2 points. Okay, so similar way we calculated distance between 2 points. So I have to cluster now similar kind of micro RNAs who could be controlled by same kind of RNA binding proteins. So what we did, these all becomes feature variables x1 x2 x3 x4 x5 करते 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 xn लास्ट तक हम लोग पहुंच गए. Okay, same way for all these feature values were compared among themselves. So imagine how many calculations we will be doing. So all these are calculated and clustering was done. So this happened. You can see the beautiful clusters happened. They formed various groups of microRNAs. Okay, and we found that most of these microRNAs which came to same cluster were being controlled by the same kind of RNA binding protein. So, but you just see that how maths is helping in the biology here. And then we reported lots of such RNA binding proteins and their role. Okay, and you can see that lots of things we have reported. Experimentally, we validated some cases here. AR2 protein, which was the highest associated bound protein we found with microRNA biogenesis. And different kinds of rishtadari, the relationship between various RNA binding protein also we defined. Somewhere antagonistic, ladai wali, dushmani wali thi, or somewhere friendship kind of thing. And we also use them to show that how good they are actually discriminating between cancer and non-cancer situation. So you can see here they were able to classify. If I use the microRNAs, those microRNAs to classify the cancer or versus normal situation, the microRNA separated them well. Okay, this is also case of unsupervised learning clustering. And here also, if the associated RNA binding proteins for these microRNA were used, the similar kind of separation were used, and very successfully cancer versus normal conditions were separated. Here you can see. So this is the implementation I'm showing you here. This is the implementation. So basically I needed data, the clipset data, which gave me this RNA protein interaction value, how much RNA a particular gene is being expressed. That value I got from RNA-seq data. How much microRNAs are being formed. That value I got from small RNA sequencing data. Then I added one more data, PPI, protein-protein interaction network data. And using all this together, I formed graphs. So graphs, what they do, किसकी किससे रिश्तेदारी है, who is related to whom, I got this network. But this network was not telling me that flow कैसे हो रहा है, किसको कौन influence कर रहा है, who is influencing who, which one. For influencing, I needed these arrows, directionality, in which direction this influence and flow is happening. So that was done using Bayesian network, BN. Bayesian network analysis was performed, and finally we got all important communications, all important paths of communications. ये क्या क्या कर रहे हैं, कौन किससे बात कर रहा है, सब पता चल गया. Okay, and this is how a particular microRNA is being regulated. So what happened here? The most important parts were used here as features. So the gene which was here, now you can see it here as a feature vector. So this way feature vector was formed in, okay, x1 x2 x3 xn तक, यहाँ तक हो गया जो कि genes थे, जो important genes. That see how much effort I had to do to derive and get those features, because I was unable to find, और अपना logic भी नहीं चल पाया था there कि भाई कौन सा genes इसमें से होगा features. So that logic worked here, and accordingly Bayesian, from using Bayesian network, those features were derived. These features were fed into machine learning algorithm that is called boosting learning, and using that one brilliant thing happened. This experiment over there, for doing microRNA expression profiling, you need to spend 2 to 5 lakhs rupees. Okay, for any given condition, you need to invest that much amount. Okay, and suppose if you have to do that kind of experiment for various large number of samples and conditions, how, just imagine how much money you will require. Now you don't require that money, you just use our models, and this is what you get, the actual versus predicted one. Our method, what our method predicted, this is the line of regression, you can see the almost accurate predictions happen. So this is the web browser we have provided on our lab website. Anybody can put here the microRNA expression data, sorry, RNA-seq expression data, and then entire system starts working like this. It goes doing decision making, then it gives result here. Then from result, you can even compare how good this result was. If you are just trying to test, you can benchmark it with your actual data also here, and it gives expression in the form of bubble chart. So bigger the bubble, the more expressed that particular microRNA was, and which gene it is controlling all these. You see which pathways were influenced, and then you get exactly look into the map which genes are regulating which part of the pathway, and which microRNA is hitting this one. Every information you get there at one place. So that is just, I'm showing you the level of accuracy, all 90 plus accuracy we attained. And then a good, beautiful example of its application. I'm showing you that we were working this thing during the COVID times, when COVID first was there. And you know that nobody was knowing what is happening within the COVID patient. There was no data. There was certainly no microRNA data also. So we used our software, and we found that first we found here in this slide I'm showing you that what kind of microRNAs are being expressed. Okay, and that data, that RNA-seq data was there. From there, we detected all these microRNAs, and then which pathways are being affected by these microRNAs in COVID, that we got detected also. You can see here. ओके, एंड ये जितना भी तुम लोग को जिंक खाने के लिए बोलता है, ये इसके कारण से खाने के लिए बोलता है. ये देखो, ये देखो, जिंक फिंगर ट्रांसक्रिप्शन फैक्टर, जिंक फिंगर ट्रांसक्रिप्शन फैक्टर, ये सब जिंक खाने का जो चक्कर है यहीं से. एंड वाई इन एक्चुअल जिंक खाने का चक्कर इधर आ गया. सो दिस इज द होल पाथवे ऑफ कोविड वी फाइनली गॉट. जिसमें जिसको कोविड होता है उसका शुगर लेवल, इट बिकम्स, द पर्सन बिकम डायबिटिक. And why the person become diabetic? Due to this one, insulin receptor. These are all affected, severely affected. This one, zinc protein, another zinc protein, all affected. And यहाँ से आपका immune system compromised here, and they here ubiquitin. These all machinery are required for protein circulation. Why body becomes weak and highly fragile? Because here, from here, zinc leakage happens a lot. So this is what we got from this entire analysis using our software. Next thing, I'm just showing you the kind of level we can get into with artificial. So how microRNA, how to detect microRNA. So detecting microRNA, in previous slide I told you how to find expression of the microRNA, how they are being formed. But if the particular region of genome is microRNA or not, that has to be defined. And here we use the latest kind of method that is called transformer method. Transformer method is like that, how you read a book, big voluminous book. You don't keep on reading is, was, that, and every word. You don't need to read every word. You need to focus on certain words, and you understand what this sentence was all about. So you, but you have, in that situation you are reading a very big motata kind of book, big book. Okay, so same philosophy is here used in transformers. So various kind of nucleotides, they are broken into different words, they are tokenized, they are given different identities, and their associations are used. So they are treated like a gene or genome sequence is treated like a book, and the nucleotide combination, various nucleotide combinations, and their structural forms were also taken into account, and they were considered as words, and how they are forming sentences, and how this sentence is studied, that is transformer. So this transformer system that studies, and finally it gives it to neural network system. Okay, and from this neural network system you get the XGBoost, and then finally if the region is microRNA or not. So we developed this system, and benchmarking is very important part. How good the system is doing, how good it is working, that has to be a certain determined. So that is called benchmarking. You compare with the existing software, that what kind of existing software are there. So I had this one, two, three, four, five software. Okay, this is my software actually. Five software with which I had to compare, and you can see that our software outperformed all of them consistently. These are the benchmarking metrics. You calculate the performance of your software and your machine learning artificial intelligence methods using sensitivity, specificity, accuracy, and FI score kind of metrics. But the most important metric is the MCC, Matthews correlation coefficient. So coming to the last point, another question was that how to find long non-coding RNAs. So long non-coding RNAs are highly variable. You can't detect them properly by the sequence similarity, and they are involved in almost everything from the DNA methylation to, you see, the transcription regulation. They work as enhancer. RNA polymerase require them to hold, because if they don't hold, the transcription process would not happen. It will go in ulta direction and thing will be another thing. So they are so much required outside the nucleus. They work like a sponge. They help in stability and translation and various things, but very less known. And humans are very well studied because we are selfish guys. We keep on studying ourselves, but there are lots of plants, and plants are equally important. And if you go into plants, there are almost no information. They are almost homology based, something like that. Okay, so plant science is very much lagging on LNC-RNAs. So here we designed LNC-RNA. So what we did, you just put your attention here carefully. Entire sequence, you see that this is the RNA sequence. So in this RNA sequence can be converted into a photographic plate. Okay. So AUGC, you have four nucleotides. So these nucleotides form the width of the snap, and sorry, the length of the snap, and the width of the snap is formed by the RNA sequence positions. Similarly, the RNA structure. So you see here the RNA structure are given in dot bracket form. I'll just show you one thing here. I kept it. RNA is not like that. What you see actually form stays in the structure form. So this is what I am just trying to show you. I run this program here. So this is the RNA sequence I paste here. Okay, and I have in what form it actually exists. That is, this is also being done by computers. This is also a sort of intelligence. So it is finding out the structure. So this sequence has actually this kind of structure. So RNA actually exists in this secondary structure form. But in computation I don't need this kind of form, so this has to be represented some way. So you see GGG, this GGG is paired, so dot bracket, it means this one starts from here and keeps on going there. Equally you read from this side, so you keep resolving internal loops, external loops. So these are paired with this one, these are paired with this one, these are paired with this one, and this region, extra remaining regions are paired with this one. Same way you can see here, you can compare it here. So RNA is converted into this structure form, and the structure information with this way extract here. Okay. Slide, yeah. So the dot bracket form you see here, this dot bracket form, and this is RNA sequence. Both of them parallelly are fed into a convolution neural network. Okay, so you see the input goes here, the input goes here, the convolution neural network starts working. A convolution neural network is followed by max pooling layer, then flattened layer, and then proper hidden vanilla layer of neural networks. Okay, and finally they are joined, both are joined together, and output is found. When output is found, our servers display the result that what which sequences had lncRNA, where the length of this is showing the kind of lncRNA, position, location, and probability values are being given. Position wise probability value is also given here, that how much chance of this region is belonging to the lncRNA. So you see here that most of them are scoring up to this part, or may say in mota moti in complete part, very highly. So they are most probably LNCRNA. Convolution neural network philosophy is like that. Anything can be represented in pictorial. It has become popular just because of your mobile phones and AI used in face recognition, things recognition. So the philosophy was basically derived from that part. And what it does, that every image is, as I told you, that it is actually aggregation of pixels. So okay, so everything can be converted into pixel. What is a pixel set? It is a matrix. So on matrix you can do computation, and you use different kind of filters. So these kind of filters I'm showing you, just showing you the filters. They keep on studying different size of filters. They keep on studying, snapping the photograph, and various kinds of dimensions are used here. Dimensions of, like this way, the vertical values, the horizontal values, they are represented this way. So all these values are given in different kind of filter layers. These filter layers are then further compressed and at very lesser information. Like earlier I was showing that, like in typical neural network, all is connected to all. Here all is not connected to all. They share weights and they are not all connected, they are treated separately. And finally all this information are fed into this neural network finally with lesser compressed information. So importance is there, that there are some features which a human can't read, it can't find out. So these features are derived by convolution neural network. So up to this step, if you look here, up to this step, sorry, up to this step, it is feature derivation problem. And once features are derived, they are inputted into the classification process, and accordingly the classification is done. So this is all about the neural network, CNN. I'm just showing how convolution is there. So what I told you, the filters are used. Different size filter, different kind of filters are used, and many filters are used. Different layers are formed, and these filters, they go there, they do some kind of linear algebra calculations, and accordingly they keep on compressing. So they go, and finally all these values are clubbed together. So all multiplied and summed, and ये जो तुम्हारा जो 9 cells था, sorry, not 9, yeah, 16, 14, 12 cells, not 9 cells, I was assuming 3. ये इमेज थोड़ा सा मेरे से गलत बन गया, but 9 cells यहाँ से है. So how many cells? 9 cells. So 9 cells calculations is done in sliding manner, and then 9 cells are converted to single cell. So compression happened. So instead of needing lots of information, you now work with much lesser information, much faster way. So this is the essence of CNN that we implemented. So filters I showed you, these are called filters. These are filters. ये इसके ऊपर से input data इस form में आता है, इसके ऊपर से it passes through, and then it outputs to the convolution layer. Okay, and then from convolution layer the max pooling layer comes. Max pooling layers आपस में nearby जो areas होते हैं, the nearby areas are calculated and summed up together and further compressed. So this was implemented by us in deep link, and we got successful result here. So this is how AI eyes, okay, our genomic system. I'm just using deep link thing here, and deep link performance is given also here, so you can see that how good deep link is doing. Deep link is outperforming all of them. All these software are outperformed by deep link by good margin. And if you come to the best metric, MCC, it is incredible. Okay, so thank you, and I welcome questions from you. You can ask me any question. Okay.
Thank you, Doctor. The session is open for discussion. If you have any clarifications, you can ask Dr. Ravi Shankar.
Any question? किसी को समझ में नहीं आया तो पूछ लो फिर से. I will go to that slide. Dr. Morgan, they are school kids, na?
College, all are undergraduate students
Undergraduate
from various disciplines of life science and biotechnology.
Okay. If you have any confusion, if you didn't understand even my first slide, let me move to that slide. I will answer you. जिसको भी कोई दिक्कत हुआ हो, समझ में नहीं आया हो, वहां चलो पूछ लो. I think there is no question, Dr. Morgan.
No questions?
Rather than I should question them.
Yeah, that can.
But I can't do. This is not open classroom. That is the problem.
Yeah.
Thank you. Then.
Thank you, Doctor, so much for elaborating the students on neural networks, mRNA, expressions.
Yeah.
Hope they understand something and they will be useful for them in the future.
Yeah, they can reach me if they are feeling shy to ask question here.
Yeah.
They should not be shy, but my lab is there. You can write to me. You can ask me any question.
I will share your email address. Maybe they can write to you if they need it.
Yeah. Okay, Dr. Morgan. Thank you.
Thank you so much.
I close it. Yeah.
Have a nice day. Thank you.
Thank you.
Okay.